We're going to take a look at some Hardy-Weinberg sample problems. The first thing that you need to know is that there are two main equations in Hardy-Weinberg problems. The first one is that p plus q is equal to 1, and p squared plus 2pq plus q squared is equal to 1. These are not ones that you actually have to memorize because they will be provided on your equations sheet that you're given for your AP test and for all of your assessments in my class. A gene is made up of two alleles. It can either be the dominant allele, the recessive allele, and any combination. So we have homozygous dominant, homozygous recessive, and heterozygous, the combination. In our equation, p squared plus 2pq plus q squared is equal to 1, we have a couple variables. The first one is that the frequency of the dominant allele is going to be represented by p. Seen here, p squared, p. And the frequency of the recessive allele is going to be represented by q every time. So if we are looking to identify the frequency of an allele, we're actually going to be looking for just p. However, if we're looking for the frequency of genotypes, so that's when we're talking about the homozygous dominant, recessive, and heterozygous, that's when we're looking for squared. So to find homozygous dominant, big A, we're looking for p squared. That's right here. If we're looking for homozygous recessive, that's q squared. And the frequency of genotypes for heterozygous is going to be 2pq. So it's important to know these and to not mix them up. You're given this top part, this equation. However, you are not provided this piece, I don't believe, so it's important to understand what these all mean. In order to have Hardy-Weinberg equilibrium, we have to follow 5 conditions. So the first one is that it is a large population, infinitely large preferably, and the reason for that is it's going to reduce the chance of disruption. The second one is that the population must be isolated from other populations. There can be no migration, so no immigration or emigration. That's because it's going to change the composition of the gene pool because someone entering or leaving the population is going to bring in different alleles. Next, there can be no mutations. Once again, that's going to change our composition of the gene pool. There needs to be random mating, and the reason for this is that individuals tend to choose mates that are similar to themselves. So phenotypically, this actually might end up the same because you're going to see the same traits expressed. However, that's actually going to change the makeup of them. And so we are going to see maybe more homozygous dominant and recessive and less heterozygous individuals. And it's important to keep that diversity in a population. Finally, there can be no natural selection. If the population is at equilibrium, no alleles are being selected for over other alleles. So these are our 5 conditions. As you can see, that's not really going to be happening in nature. But this is going to act as a model for us to compare to, and it gives us a way to calculate both allele and gene frequency in populations. Let's go ahead and do some sample problems here. So here's our first one, and I've started to put in some notes for each of them, and then as we go through each problem, I will add to it. Down here I have the same information that I had up above, but just that way we can see it in the screen. Here's problem number 1. In a certain population of 1,000 fruit flies, 640 have red eyes, while the remainder have sepia eyes. The sepia eye trait is recessive to red eyes. How many individuals would you expect to be homozygous for red eye color? So the first thing we should be doing is just taking notes on our problem. So I see 1,000 fruit flies, so I write that down. Our population is 1,000 fruit flies. How many have red eyes? Well, we have 640. Now notice that if they have red eyes, that's our dominant trait. So they can either be homozygous dominant or they can be heterozygous and still display that red eye trait. So we don't know what the makeup of the 640 is. So we're keeping that generic for now. The next thing that we have are sepia eyes, which is going to be homozygous recessive. And through 1,000 minus 640, we figure out that that is 360. So let's go ahead and start solving this problem. For problems like this, create some room here, we are really often going to be using this equation. P plus Q is equal to 1. So remember that. So the first step is going to be figuring out what Q is, because what we want eventually is to figure out how many are going to be homozygous for the red color. So we need to figure out basically what P and Q are. So let's go ahead and do this. Q there we go. So Q squared is equal to 360 individuals divided by our total individuals. So that's going to give us 0.36. So now what we can do is we can figure out Q from there because we have Q squared. So Q is going to be the square root of 0.36. So the square root of 0.36 is 0.6. So at this point, I figured out Q. From here, it's going to be really easy to figure out what P is because we know that P plus Q is equal to 1. So P plus 0.6 is equal to 1. So P equals 0.4. Okay, we now have Q, we now have P. Now remember, at that point, what that's telling us is the individual alleles, but we're not looking for just the alleles. We're actually looking for homozygous for the red eye color. So that means that P squared is going to tell us homozygous for the red eye color. So P squared is equal to 0.4 squared. So P is equal to 0.16. Now to figure out how many individuals, not just our frequency, I'm going to take that 0.16 and I'm going to multiply it by 1,000, which is how many individuals we have in our population. And that gives me 160 individuals that are going to be homozygous dominant for the red eye color. We can also use the rest of this information. We can calculate out pretty much anything that we want. Your main goal is if you can figure out Q and P, you can figure out anything for these problems. All right, here's problem number 2. Create some space. The Hardy-Weinberg equation is useful for predicting the percent of a human population that may be heterozygous carriers. So that definition of carriers is that they have some sort of a disease or any genetic indicator that is not necessarily going to be displayed, but they do have it in their DNA. So heterozygous carriers of recessive alleles for certain genetic diseases. Phenylketonuria, or PKU, is a human metabolic disorder that results in mental retardation if it is untreated at infancy. In the United States, 1 out of approximately 10,000 babies is born with the disorder. Approximately what percent of the population are heterozygous carriers of the recessive PKU allele? So we are looking for now heterozygous carriers. So once again, I did that same method. I wrote down my population. And now I have no PKU is going to be either homozygous dominant or heterozygous. That's 9,999 babies. And those that do have PKU are going to be just 1. So once again, I'm going to start with Q squared. So Q squared is equal to 1 out of, let me get out of that, 1 out of every 10,000 babies, which means that that is 0.001 0.0001. So now I want to calculate Q. This is the same method as before. So Q is going to be the square root of 0.0001, which makes it 0.01. Now I have Q, I can solve for everything else. So P plus Q is equal to 1. P plus 0.01 is equal to 1. P is equal to 0.99. Okay, Q is here, P is here. Now, if I wanted to find the frequency of homozygous dominant, I'm going to go ahead and square 0.99. If I want to find the frequency of homozygous recessive, I'm going to square 0.01. If I want to find heterozygous, now I'm going to use 2pq. So that's going to be 2 times 0.99 times 0.01. Go back here. So that means it is going to be let me do that real quick. 2 times 0.99 times 0.01 is 0.0198. So now to find the percent, I'm just going to go ahead and move my decimal over 2 places to the right, 1, 2, and that gives me 1.98%. Now because it's asking approximately, I'm going to go ahead and round that. So that means it is going to be approximately 2%. And that is my final answer. Once again, we have Q and we have P, so there's a lot of different things that we can figure out through this way. We're going to move on to a little bit more difficult of a problem. Let's move this down to the next page. Okay. A very large population of randomly mating laboratory mice contains 35% white mice. White coloring is caused by the double recessive genotype, so a, or homozygous recessive. Calculate the allele and genotypic frequencies for this population. So if it's being generic like this and saying calculate the allele and genotypic frequencies, we're going to need to do that for all of them. So let's go ahead and start with what we know. We know, let me do this up here, that we have 35% is going to be white mice. We know that that is going to be a, which is equal to Q squared. Okay, so Q squared is going to be equal to, if we just make this into a decimal, 0.35. In order to find Q from here, we know we have Q squared, so all we have to do is take the square root of 0.35. The square root of 0.35 is going to give us this answer, which is 0.59. So once again, we've identified what Q is. Using P plus Q is equal to 1, we know that P is equal to, I'll write it out for you guys, P plus 0.59 is equal to 1. So now we know that P is equal to 0.41. Okay, so we have Q, we have P. That is all we need to know now. Moving forward, we can solve everything else. So we have our allele frequencies. That's located right here. Now we need to find our genotypic frequencies. To find our genotypic frequencies, we're using this. So let's go ahead and start off with big A, which is equal to P squared. That means we're going to go ahead and figure out P is right here, so that's 0.41. Square that. So that is going to equal 0.17. That's our first answer. Let's highlight that in a different color. All right, next one. Let's figure out heterozygous. That's our next genotypic frequency. Go back yellow. Oops. Big A, little a is equal to 2 PQ. Oops. Still want that one to be green. There we go. Okay. 2 times P, which is 0.41, times Q, which is 0.59. That is going to give us the answer of 0.48. Finally, let's find our homozygous recessive. Oops. All right, homozygous recessive is simply going to be Q squared. All right, Q is 0.59. So let's square that. And that gives us 0.35. Okay. Now, let's check our work. So this is when we're going to use this equation here to make sure that we have the right answer. And I'm going to highlight that one in green. So what we've calculated thus far is we have figured out that homozygous dominant is P squared. We squared P, 0.17. Heterozygous is 2PQ. We multiplied that out. We got 0.48. And we figured out that homozygous recessive is Q squared, which we did here, and that gave us 0.35. So now what I'm going to do is I'm going to just plug all of those in. So P squared, 0.17, plus 2PQ, which is big A, little a, that's right here, 0.48, plus little a, which is Q squared, 0.35, and that should equal 1. So let's add all that up. 0.17 plus 0.48 plus 0.35, and sure enough, that gives us 1. So that's a way to verify our answers. So now we know for a fact that that is correct, or at least it seems to be correct. To answer this question, to write it out, the allele frequencies are going to be right here, Q and P. And then our genotypic frequencies are the ones highlighted in green here that we just verified in this equation. So as you can see through these problems, what's really important is knowing P plus Q is equal to 1. If you can calculate out Q, you can easily find P. From there, you can plug it in and find anything out. Realize that you don't always need to put it into this full equation. It's more just to check your answers. You can actually just use this right here to figure out what the question is asking for. It's just important to pay attention to what information you were given and how you can put that together to solve your problem.